So the FDA has recently issued 6 new guidance documents in the field of cell and gene therapy. The most significant one, in my opinion, is a revised CMC guidance. There are 3 disease-specific guidances, a guidance on long-term patient monitoring, and a highly focused guidance on retroviral testing. We are somewhat disappointed with the CMC guidance. The main change in the CMC guidance is its reformatting into common technical document format. The amount of genuinely new content in this guidance is very small. Obviously, we and the rest of the field are regretful that 10 years has elapsed since the previous cell or gene therapy CMC guidance. We congratulate the agency and the new Commissioner, Scott Gottlieb, on issuing a new guidance, but we very much hope it will not be another 10 years before further guidance in this area. The field of cell and gene therapy has moved faster in the last several years than in the last several decades. This is acknowledged by the agency in these guidances, but we feel it is not yet reflected in actual content. We are still looking for more specific guidance, and we would like to see more specialized guidance to focus on pure play cell therapies, which are not gene-modified cell therapies, and pure play gene therapies, wherein the cell is relegated to a manufacturing platform rather than being the product itself. We feel that each of these 3 categories deserves a specific guidance of its own, and we very much hope the agency will follow through with such guidances in the near future. Something unique to the field of cell and gene therapy at this point in time is an increasing number of licensure events from earlier and earlier clinical trials. In the face of efficacy levels such as seen in the Eliana trials and the Zuma trials for Yescarta and Kymriah, licensure from phase 2 is going to become increasingly common. Unfortunately, phase 2 clinical trials are rarely supported by commercial grade CMC. We strongly enjoin the FDA and other regulators to provide, if you will, a punch list of must-have CMC measures which must be put in place in order to support licensure. The current guidances are not clear in this way and honestly are not designed to support licensure from such an early point in the clinical development process. We feel that CMC measures which are not included in the punch list can readily be backfilled post-license by post-licensing commitments to make a complete CMC panel of measures taken at some point during the product's trajectory, but not necessarily at the exact point of licensure, especially if it comes out of a phase 2 trial. We think this is an absolutely essential measure that the agency should take. We sense reluctance of the agency to commit to these measures, and we hope that we can help them change that as soon as possible. Everybody wants to get approval out of phase 2. Out of our phase 2 trial, you will risk falling behind your competition. This is something which is increasingly happening across the entire field of medicine, but it's also happening disproportionately in the field of cell and gene therapy. I think phase 1, phase 2, phase 3 will become a thing of the past. Scott Gottlieb has vigorously promoted the concept of seamless clinical trials, where we avoid the phase 1 stop, phase 2 stop, phase 3 scenario. And I think this kind of radical measure in clinical trial design is outstanding. We wish we saw the same level of enthusiasm for reforming their CMC guidances. The FDA's duty, in addition to approving drugs, is to protect patients. And this does not naturally set up an atmosphere which is conducive to a deep dialogue between the pharmaceutical industry and regulators. We feel at this point it is hard for the FDA to approach pharmaceutical companies and biotech companies directly for input, but the onus is on those companies to come forward with their own suggestions for improving the guidance. Dark Horse feels uniquely positioned to do this because of our wide client base, and we hope that we can integrate our clients' responses and our clients' wishes and needs and provide a cohesive input to the agency for improvement of these guidance documents. One area where we are going to provide input is the division of the current single CMC guidance into multiple CMC guidances. We feel that the product diversity in the field, pure play cell therapies, gene-modified cell therapies, pure play gene therapies, the diversity in the field no longer justifies a single guidance, because aspects of, for example, a pure play cell therapy guidance are not in any way relevant to a pure play gene therapy guidance. So we're strongly recommending to the FDA that the guidances be subdivided by product type, which will enable them to be deeper and more specific to the various products they address. As soon as a draft guidance is published, the FDA will accept any feedback at any time on that document. It's a legal possibility to submit feedback at any time on any guidance, whether or not it's in draft or it has been finalized. We think that these documents must be living documents, and we very much hope that a lot of feedback will continue to be provided to the FDA because the field is moving so quickly. The first guidance in 2008 obviously is tremendously outdated. The new guidance does not offer materially different guidelines for very many technical issues. It's mainly a reformatting exercise to enter CTD format, but we hope that the agency receives a lot of feedback over an extended period of time and that these documents are updated very often.